Welcome to Grade 12 Life Science. This is a first in a series of lessons on life at the molecular, cellular, and tissue level. This lesson focuses on DNA, the code of life. Life itself is interesting, and understanding the code of life is a bit more exciting. All living things are made up of cells. Plants have plant cells, and animals have cells that are slightly different to that, called animal cells. But looking at cells, the most conspicuous thing that we see in cells are the nuclei. Initially, the cells had organelles without membranes, and these cells were called prokaryotic cells. Cells later on had organelles covered with membranes, and these became eukaryotic cells. Let us understand the nucleus. The function of the nucleus is firstly, to control all the activities of a cell, and the second function of the nucleus is to transfer information from the parent to the offspring. Now to recap material that was covered earlier on in Grade 10, the structure of the nucleus includes, firstly, the nuclear envelope. Now the nuclear envelope surrounds the nucleus, and they are broken up by nuclear pores. These are little gaps in the nuclear membrane. The material inside the nucleus is called the nucleoplasm, and within the nucleoplasm, we find there's also a mass of threads that we refer to as chromatin material. This chromatin material, when separated, can show the presence of chromosomes. Lastly, within the nucleus, we may find one or more nucleoli, and these nucleoli store RNA. Chromatin material is made up of chromosomes. Each chromosome comprises of a double chromatid joined by a centromere. The chromosome itself is made up of DNA strands, and these strands are wrapped around proteins which we refer to as histones. Short lengths of DNA are called genes, and they code for characteristics. For example, you may have a gene on one chromosome for eye color, you may have a gene on another chromosome on that position for shape of ears. The genes are small segments of DNA. When we consider DNA, we find there are three types of DNA found in a cell. The nuclear DNA, that is found inside the nucleus in the chromosome. Chloroplastic DNA, which is found in the chloroplast, obviously in plant cells. And mitochondrial DNA, which we will study also in a later section on evolution. DNA actually looks like this. And it took a long time for them to understand how DNA looked. It was the work of Rosalind Franklin, Maurice Wilkins, James Watson, and Francis Crick that took a lot of time and experimentation to discover the structure of DNA. Watson and Crick received the Nobel Prize for their work. Rosalind Franklin was later honored for her contribution. She had passed on when they had made this presentation to Watson and Crick. When they described the structure of DNA, they described it as a double-stranded helix. These are the main guys that were responsible for finding out the structure of DNA. When they had completed their work, this is the structure that they proposed. The structure of DNA. DNA stands for deoxyribonucleic acid. Being a large molecule, or a polymer, it's obviously made up of smaller units, monomers that we call nucleotides. Each nucleotide has a phosphate, a sugar. I'm writing the symbol D, because the sugar is deoxyribose, and a nitrogenous base. Four nucleotides have been identified in DNA: adenine, thymine, cytosine and guanine. They've been symbolized A, T, G, and C. Whenever you see the symbol A, it stands for adenine. If you see the symbol T, it stands for thymine. If you see the symbol C, it stands for cytosine. And if you see the letter G, it stands for guanine. You may use these letters in your examinations. Adenine and thymine, A and T, form one base pair. That means they are linked. Guanine and cytosine form a second base pair. The base pairs can be seen in this diagram, guanine and cytosine. Or cytosine and guanine, that is one base pair. Adenine and thymine, or thymine and adenine. You will not find adenine joining to guanine, or cytosine. You will not find guanine joining with thymine. They will stay with their base pairs. Adenine and guanine are very long molecules, and they are called purines. Thymine and cytosine are called pyrimidines, and they're much shorter molecules. The DNA molecule resembles a twisted ladder with the sides of the ladder being made up of phosphate and sugar. Phosphate, sugar. Phosphate, sugar. Phosphate, sugar. And the same applies on this side. So, the sides of the ladder are made up of phosphate and sugar, whereas the rungs of the ladder, this area, is made up of your nitrogenous base pairs. DNA replication. What is DNA replication? It involves the making an exact copy of the DNA. When does DNA replication take place? It actually takes place just before a cell divides, a time that we call the interphase. Why does DNA replication take place? Well, it takes place to ensure that daughter cells that are formed are identical to the mother cell. All these changes that take place during replication takes place in several steps, each of which are controlled by enzymes. Your exam requires you to understand the process of replication and to be able to describe in detail, step by step, how this process occurs. Let us take a look at this process. It begins with the DNA unwinding. If that's our DNA, it now unwinds, and this would make it look like a ladder. This is obviously controlled by enzymes. The next step is it unzips. To unzip, it has to break the weak hydrogen bonds. Again, a stage controlled by enzymes. When the strands have unzipped, you land with two single strands of DNA. Each of these strands now act as a template to form new strands. Free nucleotides from the nucleotide pool position themselves in complementary positions along the templates. Adenine joins with thymine, guanine will join with cytosine, and vice versa. When they join, they form weak hydrogen bonds. The phosphate and sugar then join, and lastly, two new strands are formed. They are identical to the original strand, and they are identical to each other. So basically, we have made exact copies of the DNA. The following slide shows us exactly how this would look, giving a little bit more detail about the process. There's our initial chromosome containing DNA. Here, it unwinds. Here, it unzips. These are our two strands, 1 and 2. And these are our new strands being built. Remember I said, each strand is identical to the other, and each strand is identical to the original strand from which we started. To summarize, from one DNA strand, we produce two identical new DNA strands, so that is basically DNA replication. DNA profiling, or DNA fingerprinting. It's actually not the same thing as taking fingerprints or thumbprints. DNA profiling is a biotechnical process that used blood, saliva, hair, or semen. During this time, an individual's DNA is fingerprinted using fragments of DNA and X-ray technology. These appear as a pattern of dark bands. DNA profiling is unique for all individuals, except for identical twins. And then, the suspect's profile is matched with evidence. Here is an example, similar to one that you may get in an exam. Let us read through this case study. Inspector Ndlovu and Sergeant Boda were investigating the scene of a violent crime. The victim of the crime was a 70-year-old woman. She had been stabbed and left to die. They found a few pieces of hair in one of her hands, which might have come from the murderer. There was also skin under her nails, which she could have got from the murderer, if she had been able to fight back. There were 3 possible suspects in this investigation. All 3 suspects were required to give blood, and a sample was also taken from the victim. DNA fingerprints of the 4 samples were compared with the DNA fingerprints of the 2 samples taken from the crime scene. Here, we have DNA from the hair, DNA from the skin. Here, the victim, and the 3 suspects. The question asked, was the DNA from the hair and the skin under the nails of the victim from the same person? In order to understand this question, and to arrive at an answer, you need to look at the bars that are on these DNA profiles, and then try to match them. In answering the first question, was the DNA from the hair and the skin under the nails of the victim from the same person? The answer would be no, so number 1, no. In order to elaborate, one would then say that the bars don't match. Question 2. Could any of the 3 suspects have been present at the crime scene? Give a reason for your answer. Once again, you need to compare the DNA profiles. Have you come up with an answer? You are correct. Suspect 2 seems to be at the crime scene, because the DNA profile of Suspect 2 seems to match the DNA profile of the hair. Question 3 says, compare the DNA from the skin found under the fingernails of the victim with the DNA of the victim. What do you notice? Skin with the DNA of the victim. I'm sure you would agree that the bars don't correspond or match, so in answering this question, we would say, compare the DNA of the skin found under the fingernails of the victim with the DNA of the victim. What do you notice? The answer would then read, there is no match. Meaning, the bands do not correspond. To question 4, do you think DNA evidence on its own is enough to convict a criminal? So, they are asking, if we had these profiles, and the profile matched number 2, as this case, would we be correct in stating that the suspect 2 was the murderer? Well, unfortunately, we cannot say for certain that suspect 2 was the murderer. What would be my reasoning? Well, put in another way, someone could have planted this DNA sample in the scene, and thus frame suspect 2. The answer to this question Do you think DNA evidence on its own is enough? No, and the reason for this is that the DNA could be planted at the scene, and this would then frame a certain person. The last question. Do you think it would be a good idea to collect the DNA from every person in the country to create a DNA fingerprint database? Provide arguments for and against. You may provide more answers, but we will just take one for and one against. Why would it be a good idea to have the DNA fingerprints of everybody in a country? Say yes. It will be easier to identify criminals. Absolutely. If we have a crime scene, and we have some DNA, we just go through our database, and then we can match the DNAs. If we had to look at an argument against it, why won't it be a good idea to have the DNA of everybody on our database? First answer is that it's too expensive a process, and also a very good answer would be that our DNA information is basically our personal information, and we have a right to revealing our personal details. Uses of DNA profiling. Firstly, like we have done in the previous slide, it's used to identify criminals. Secondly, if siblings are lost at birth or separated, you are able to identify the siblings. In order to diagnose genetic disorders, paternity testing may be in families to identify the father. And also, to use human remains at an accident scene or at a site to provide positive identification to identify human remains. However, this method may not be totally acceptable in a court of law. Why would this be so? Simply because there is no uniform standard procedure for taking fingerprints, or DNA fingerprints more correctly. If everybody in all the countries, and all the different towns were using the same technique, it may become more reliable, but we are unsure about the standard, or the procedure that they are using. And secondly, it is possible to plant DNA, and to frame people at a crime scene. The Human Genome Project. Genome is the exact DNA sequence of an organism. The Human Genome Project was completed in year 2000, at the turn of the millennium. It mapped out the position on every gene in the human race. The genome of other organisms have also been done. It is interesting to note that there is only a 2% difference between the human and chimpanzee, yet we are so much different in appearance and behavior. The other nucleic acid that we want to teach in this topic is called RNA, and RNA stands for ribonucleic acid, symbolized RNA. Being a polymer, it's also made up of monomers called nucleotides. The bases present in RNA are similar to those in DNA, except thymine is replaced by uracil. So the bases present in RNA would then include adenine, guanine, cytosine, and uracil. Structurally, RNA is a shorter strand, and the sugar present in RNA is ribose. 3 types of RNA can be identified: messenger RNA, symbolized mRNA, found in the nucleus; ribosomal RNA, symbolized rRNA, found in the nucleolus, and at a ribosome; and transfer RNA, symbolized tRNA, found in the cytosol. Listening to the description that I gave of RNA, are you able to compare the structure of DNA and RNA? Do it in tabular form, that means, draw it in a table. The differences between DNA and RNA. You may take a few minutes to do this. Did Your answer look like something like this. If you're looking at DNA, on the left-hand side, and the structure of RNA, the first comparison would go on the lengths of the chain. In DNA, the chains are long, so a long chain as compared to a short chain in RNA. A double strand in DNA, a single strand in RNA. A spiral helix in DNA, no spiral helix in RNA. The sugar in DNA, called deoxyribose. The sugar in RNA, called ribose. The bases found in DNA, adenine, thymine, guanine, cytosine. The bases found in RNA, adenine, uracil, guanine, cytosine. In DNA, base pairing occurs. In RNA, a single strand, no base pairing. So this would be the answer to tabulate the differences between DNA and RNA. You may get a mark for drawing the table as well. This topic deals with protein synthesis. Almost 80% of our body, that means the dry mass, is made up of proteins. Proteins are large polymers, and they are made up of monomer units called amino acids. If these blue little circles represent amino acids, this could be amino acid 1. This could be amino acid 5. This could be amino acid 17. This could be amino acid 11. So, a series of amino acids joined, one to the other would then form a protein. In order to call the compound, or molecule, a protein, you must have more than 50 amino acids. From earlier knowledge, you will understand that enzymes and hormones are protein in nature. Protein synthesis occurs at the ribosome, a structure found in the cytosol of cells. Let us recap. Structure of the nucleus. On the outside is the nuclear membrane. Some books would refer to it as a nuclear envelope. We might find a number of strands, either collectively or singularly spread out in the nucleus, and this would then refer to your chromatin, or individually as chromosomes. This little dot indicates a hole in the membrane, so that will be your nuclear pore. And the denser structure within the nucleus would then be your nucleolus. Everything inside the nucleus that is indicated in blue would then be nucleoplasm. To understand protein synthesis, we need to break the process up into two stages. The first part is transcription, and it takes place in the nucleus, the second part being translation, which takes place in the cytoplasm. Firstly, transcription. During this process, the DNA, which was a double helix, unwinds. Very similar to that in replication, and then it unzips. Now, in DNA replication, both strands were copied, but in protein synthesis transcription, only a single strand is copied, and we form messenger RNA. So, this is messenger RNA. It was formed from copying one strand of DNA. In order to form this mRNA, free nucleotides from the nucleotide pool was used to form the messenger RNA. Messenger RNA then leaves via the nuclear pore. When it leaves the nucleus, it goes to the cytoplasm. The second stage in protein synthesis is called translation. This is obviously going to take place outside the nucleus, and it's going to occur at the ribosome, the site of protein synthesis. At the ribosome, the messenger RNA straddles itself around the ribosome. In order to join the ribosome, it uses rRNA, ribosomal RNA. The next step is that transfer RNA, symbolized tRNA, aligns itself along messenger RNA. It does so by using the 3 bases along the messenger RNA that we call codons. So, each 3 bases that are consecutive constitute a codon, and they're actually code for a particular amino acid. Each transfer RNA has a triplet code, and this triplet code is referred to as the anticodon. So, on the messenger RNA, you have codons. And on the transfer RNAs, you have anticodons. Once the transfer RNA positions itself on the messenger RNA, the amino acids are then sequenced. The amino acids then join to each other by peptide bonds to form your protein. The following is part of an examination paper, just to practice what we have learned. If I consider this diagram, it says, the diagram below shows a part of the process of protein synthesis. When I'm studying this diagram, I need to identify firstly, ribosome. Molecule X, single strand, molecule Y, I see letters all over. The question asks, name the stage of protein synthesis, represented in the diagram. To answer this question obviously, I can see ribosomes, so the answer to that is cytoplasm. So if it's cytoplasm, then translation. The next question asks you to identify molecule X. So, molecule X, like I identified earlier on, was just a single strand, so it could only be mRNA. Molecule Y, if I look at it closely, shows 3 bases on one end, and amino acid at the other end, so molecule Y would then be tRNA. And then, the last question says, identify structure 1. Now structure 1 is talking about those triplet bases that are present on molecule X. So structure 1, molecule X, let's think again. Molecule X was messenger RNA, so the 3-letter bases on molecular RNA is codon. Well done. That would show the type of questions on protein synthesis.